again, we don't have Phil here, but we do have Phil here. I feel very fortunate, he's in Virginia. And Phil and Eric have been friends and colleagues for a number of years. And so if everything goes correctly, we're hoping, so just keep the voice down, Phil is going to, and there's Phil, wearing his PSB hat, an old one. We are sending him a new one. He's not being cheated out. But Phil is going to provide the first 2/3 of the introduction for Eric. So Phil, hopefully this will work. Go for it.
Okay, thanks, Terry. Can you hear me okay?
Yeah.
Yep, all
Well,
good.
First I want to say, it's only health reasons that are keeping me away. I think I've attended 27 of the 31 PSBs. It's been a very distinct part of my academic life, and I'm very sad not to be there. But that aside, let me introduce Eric. Eric Green, MD, PhD, has been a prominent member of the genomics research community since the inception of the field in 1987. As part of the Human Genome Project, he played a central role in the mapping and sequencing of the human genome and has since served as an international genomics leader. Eric received his BS in bacteriology from the University of Wisconsin in '81, his MD and PhD from Washington University in '87. Coincidentally, in '87 was the same year that the word genomics was coined. During residency training in clinical pathology, laboratory medicine, Dr. Eric worked in the laboratory of Maynard Olson, where he launched his career in genomics. As a physician, he is drawn to genomics because of the potential of using patients' genomic information for improving their medical care and management, something I'm very happy to have right now. Prior to joining National NHGRI in '94, he was assistant professor of pathology and genetics, as well as a co-investigator in the Human Genome Center at Washington University from '92 to '94. As NHGRI Director for 15 plus years, Dr. Eric led a government agency with an annual budget that had $650 million and a staff of around 640 near the end. Under his stewardship, the Institute completed 2 cycles of strategic planning to ensure that its research investments in genomics effectively advanced human health. The first effort yielded the highly cited 2011 strategic vision, charting a course for genomic medicine from base pairs to bedside. And the second yielded the strategic vision for improving human health. Eric has received numerous awards, including being elected to the National Academy of Medicine in 2023. Eric retired from federal service. He looks, I have to say, he's looking at the cap era all the time, and is now seeking opportunities to use his extensive experience in leading government-funded research programs to foster the expansion of genomics in academia, industry, and everyday life. If I might just add one quick comment. Eric was an absolutely amazing mentor to me when I went to the NIH to be the first Associate Director for Data Science at the NIH. And we wouldn't have got anywhere near accomplish what we managed to accomplish if it wasn't for his experience and savvy across the NIH and across the sciences. So thank you. Terry, I'll leave you for the funny parts.
So, thank you, Phil. So I just want to add on a little bit. First off, we're very grateful for Phil for Zooming in. It is not easy to get someone from Virginia. This is why we don't do typically live presentations. And so, Phil, thank you for all the testing we did, and thank you for the patience. And Eric, if you couldn't hear some of it, I do have a lot of it written down. I just want to add a few things. First off, the one award I'm really jealous of is that he is in the National Academy of Medicine. I'm not interested in awards, but that is one award that I would have liked. But really what I want to tell you is how I got Eric here. And it was a bit of a surprise. So on March 21st, there was a public announcement from NIH that said, and I'm quoting, "Dr. Green's tenure ended on March 17, 2025, and his appointment was not renewed." Now, a number of us had already heard this through the grapevine, and a number of us were rather sad about this, to say the least. And we couldn't act upon that information. It's just, that's just the way it works. But after they announced it, on Sunday, March 23rd, I sent Eric the following message on behalf of the PSB co-chairs, because, I don't know, I got it in my brain that I thought, oh, wouldn't it be cool? So I'm just going to read it to you. Be patient. Dear Eric, because part of it is more important. First, I want to express my gratitude for your vision, leadership, and passion that you have shared with the world. I know I am just one of thousands who appreciate everything you have done and are deeply disappointed at the current situation. And for transparency, I do have several grants through NHGRI. And that was important to me to share that. I think this is important for everyone who is a scientist right now to share with anyone at NIH your appreciation for the fact that either they were there or they're still there fighting on behalf of us. This is critical. It is absolutely critical. So, and at NSF, et cetera. So please show your support, be patient with these people, and show your appreciation. Anyway, I said, now for the second less important part of this email, but perhaps timely. My understanding is that you will not be at NIH come January 2026, which means you are able to travel. As you may be aware, 30 years ago, Larry Hunter and I began the Pacific Symposium on Biocomputing, known as PSB. Over time, we expanded that leadership to include Marilyn Ritchie, Russ Altman, and formerly Keith Dunker, who's now emeritus. PSB is an international bioinformatics computational biology meeting that has been held annually since 1996. And then I had a whole bunch of stuff about PSB. I'm under the belief that Eric had no clue who I was, just to be clear. In fact, I copied Aaron Ramos, who is the Deputy Director of Genomic Medicine, and Terry Manolio, the Director of Genomic Medicine, just so for credibility, in case he didn't know who I was. Now, it turns out Eric does know who I am, but, you know, I suffer from a few things. Okay, so anyway, I said I have copied Aaron and Terry on this email in the hopes that that they will raise this email to your attention, as I'm positive you must be getting thousands of emails daily at this point, and because they are both familiar with the excellence of the meeting, having held workshops and a keynote related to ClinGen. If you look at the previous meetings, papers, and attendee list, you'd be amazed at how much your influence has been over the years. And I hope you'll be able to come. And that's something that's really critical. If you look back over the 20 years, I cannot overemphasize Eric's leadership and guidance has had such a huge effort here. I recognize at this point of time that it might be difficult for you to commit not knowing your schedule and what you'll be doing. However, if you know it isn't an immediate no and not of interest to you, would you please let me know in the next couple of weeks? We kind of work fast on keynotes. I understand that you may not be able to commit until summer, for example, and I'd be happy to discuss this with you on the phone if that would be useful. I can guarantee that the venue cannot be beat and the quality of the science is very high, and hopefully, Eric, that is what you're finding. And again, Eric, thank you. We will miss you greatly in your leadership role with the government. I hope that in some fashion you will still share your sense of humor, insights, and vision, even if it is while sitting by the side of Snake River in Idaho or Wyoming and watching the water go by. 18 hours later, I'm not joking, 18 hours later, I received the following. Thank you for your kind words and the invitation. I have immense flexibility in my life now, so count me in for this keynote talk. Russ Altman can tell you I was texting like, you can't believe what I just did. Anyway, so we're very fortunate to have Eric join us today, and with a warm welcome, I'm delighted, Eric, coming up to present you with this lei as a symbol of aloha and welcome to the PSB community. We hope this will be one of many PSBs that you will attend. I'm going to put this on you. Feel free to take it off afterwards.
No.
But welcome.
Thank you. Well, thank you to Phil. Phil, I hope you're still there. I really appreciate it. Very kind words, Terry. That was a fantastic introduction. Of course, my reaction to it is I don't even know what I was doing for those 18 hours. What I was ready to respond immediately. Nothing like unemployment to give you plenty of time to travel or to see various things. But I'm thrilled to be here. It is the first time I've been at this meeting. I don't know what I was doing for the previous 30, because I'm very impressed with everything I've seen, and I've enjoyed the pre-meeting workshops yesterday and the session this morning. And so thanks for the invitation. By the way, you can photograph or tweet or do any social media. I know the speaker's supposed to say if they want to lift those restrictions, lift away. You can do anything you want. What I want to do today, and I think it is very appropriate relative to the nature of the invitation, is really just to tell you a story. I'm going to tell you the story of human genomics from the beginning up until the present time. It's very appropriate for the session that we were just a part of related to precision medicine. And I really hope to capture the imagination of the very heterogeneous group of people that are here. I see people from several generations. So some parts of my story are people are going to, it's like a walk down memory lane of the early days, and other people you've only heard about the Human Genome Project in the history books. And so I think there's going to be a little bit of everything for everyone. Now, I did also want to interject one other thing about, maybe say a little bit of something about myself. Maybe people knew about this, or maybe they didn't, and I want to make sure I can advance this. Okay, so I trained as a physician scientist, similar to Anne, our first speaker from this morning. My training took place at Washington University in St. Louis. And I, in so many aspects of my life, feel like I was incredibly fortunate. I was always seemed to be in the right place at the right time, and many of my successes, really all of my successes, really link back to specific parts of my academic and educational upbringing. I think Washington University was a great place to train. Choosing to get an MD PhD I think was a great decision. But I was also blessed along the way by having incredible mentors that really shaped me and prepared me for what I eventually had the opportunity to participate in professionally. And ironically, the other thing is, which maybe people don't know about, is you can't imagine how undertrained I was in genomics when I got involved in this field. Because as a graduate student, I had nothing to do with molecular genetics or molecular biology or anything. But I am fortunate today to have, sitting in the audience in the fourth row, one of my two PhD advisors. This is Jacques Benziger, who's in the fourth row. And it was actually a black and white photo, but it made us look so old that I actually color enhanced it with AI yesterday. But I was his PhD, it was Jacques and another gentleman named Irv Boyne, but I really spent the majority of my time in Jacques' lab from 1983 to 1986. That was back in the days when MD PhD students actually could do PhDs in 3 years. Don't get me started. I think that's one of the problems with MD PhD programs these days. But it was an incredible experience. But what did I work on? Well, I actually worked on sugars and carbohydrate type things, but they weren't simple linear things. Jacques and I would always say that DNA was pretty boring because it was linear. We worked on these chains, asparagine-linked oligosaccharides. This was sort of the first paper I published as part of my PhD, where Jacques and I elucidated the structure of asparagine-linked oligosaccharides on a set of glycoprotein hormones. Jacques and I never said the word genomics once in that time because, as actually you heard from the introduction from Phil, the word didn't even exist. And in fact, I will remind you that when I graduated with my MD PhD, which was 1987, was actually the year the word genomics was even coined. But I did have this, now why is Jacques in the audience? I don't take him to all of my meetings. It's just ironically 20 years ago, he and his wife retired, and they live 1.5 miles up the road here. And when he heard I was coming to this meeting, he said, I'll just mosey over, I want to hear your talk. So Jacques, thank you for everything you've done for me over the years. And it was really all part of great mentorship I had, starting with Jacques as a PhD student, later Maynard Olson, and so forth. So. But now let me get back to the story. And the story I really want to tell you, I would contend, really relates to things that were started last century. I would contend that two interrelated scientific fields launched last century really are changing medicine this century. We heard about it all this morning. Many of you may not realize genetics as a discipline, as a word, got coined and got started in the early part of the century, 1907. I would contend the halftime of last century was when the scientists discovered the double helical structure of DNA. In 1953, and then in the year I graduated with my MD PhD degree, in this paper, which put the word genomics into the scientific literature for the very first time in this lead editorial of this new journal called Genomics, this is when genomics actually come to pass. I was very fortunate because I had just graduated that year. I was training in clinical pathology, and I had an opportunity to then work into a genomics lab that just was, the word had just been coined, and therefore was able to participate in the early stages of the field. Now, before we even got there, let me just remind you that following the discovery of the double helical structure of DNA came a series of seminal discoveries that really started progress towards better understanding how it was that DNA was the information molecule for all living systems. We elucidated the genetic code. By the 1970s, we start to figure out how to read out the DNA letters. And then, of course, the recombinant DNA and molecular biology revolution came, so that it allowed all sorts of methods for manipulating DNA, cloning DNA, amplifying DNA, and so forth. What this allowed molecular biologists, molecular geneticists to do was to basically start to develop a tool belt filled with better and better tools that would be available for studying DNA. And so when it came to the early part of the 1980s in particular, there started to be a drumbeat of discussions leading up to the Human Genome Project. Again, a walk down memory lane for some people and a history lesson for others. The first seminal meeting where this was discussed was a meeting in 1984 called the Alta Summit, where they brought people together to talk about the idea of maybe comprehensively studying, elucidating the sequence of the human genome. Renato Del Becco, a prominent cancer researcher, wrote a very influential editorial in 1986 saying that if we were ever going to understand cancer, we needed to sequence the human genome. That drumbeat led up to the naming of the field in 1987, as I talked about. But then two influential scientific groups got together, one under the auspices of the National Research Council, one under the auspices of the Office of Science and Technology Policy associated with the White House, and in 1988 put out these two blueprints, if you will, for what eventually would become the Human Genome Project. Of course, that required money. Of course, that required leadership. And then in 1989, a key Senate hearing was held, and that really formulated and authorized and then appropriated funds that allowed the Human Genome Project to get out of the gate in 1990. I believe that 1989 will go down in history as one of the most significant years, especially in terms of scientific inquiry and other important societal developments. 89 will just be remembered by the world for generations to come. Because in 1989, everything really started to change. You know, first of all, Taylor Swift was born, okay? So we should all acknowledge that is just seismic in its influence, okay? And along the way came the late planning for the Human Genome Project. So 89, these two images forever, you should remember them. 89 was a pivotal year. Like I said, I was so fortunate. Washington University had all the right pieces in place to get one of the first funded genome centers, so that there I was able to participate along with the other centers that were funded in going and launching the Genome Project in 1990, which really involves reading humanity's blueprint. I'm not going to go through many details about the Genome Project other than to say that along the way there were some important historical milestones prior to the completion. Some of you might have heard about, and by the way, the other thing about genomicists, we love odometer moments. We love to kind of celebrate them every opportunity. So last year was the 25th anniversary of the race between Celera Genomics and the Human Genome Project was declared tied with President Clinton residing over this announcement. And I'll just warn you, because then those groups went off and published two papers, the Human Genome Project paper in Nature, reporting the draft sequence of the human genome, and the Science paper that featured the Venters and Celera Genomics draft sequence of the human genome. So be prepared, because now it's going to be the 25th anniversary of these publications, and I already know of multiple events and multiple journal publication highlights to commemorate the 25th anniversary of the draft sequence of the human genome. Of course, the Genome Project itself didn't get completed until 2003. It was ahead of schedule. It was originally thought to take 15 years, only took 13 years, and had accomplished essentially every goal it had set out to do, which is why the project ended early. And so that by 2013, and having completed the signature goal of the Genome Project, that is to generate the first near complete sequence of a human genome, the project was declared completed. Major press releases announced its completion of April 2003, lots and lots of parties and celebrations, and there we were having the 3 billion ordered letters of the human genome to read and to interpret and to constantly peer over, as we are doing up to the present time. So that's a quick romp through that historical circumstance that leads us to now. It's almost 23 years ago. But of course, let's keep in mind that the Genome Project was by no means the final destination. It was certainly a finish line, but that finish line quickly became a starting line. And so much of what we've talked about this morning in thinking about precision medicine really reflects how the Genome Project just was the start of everything, even though we recognized it as an important finishing accomplishment of this first journey, as I often describe it, associated with the field of genomics. A lot has happened, and a lot gets discussed at this meeting, and a lot of areas of life sciences have benefited tremendously from the accomplishments of the Genome Project and what's gone on since then. I was at the National Human Genome Research Institute, created by the US Congress to lead the US's effort in the Human Genome Project, but of course, having accomplished the goals of the Genome Project, 23 years ago, it became clear that that institute was going to need to sort of pivot to have a new mission. But it was pretty clear that it was embedded at the National Institutes of Health for a reason, because it was widely regarded that if the Genome Project would be successful, it would set up an opportunity to use genomics to improve the practice of medicine and eventually advance human health. So for that reason, it wasn't terribly surprising that as soon as the Genome Project ended, whether the popular press, New York Times Magazine, the scientific press, they put the two words together, genomic and medicine. It was the obvious thing to create this phrase called genomic medicine. And that was great, and it was wonderful. There was only one problem with it, is that we could spell genomic medicine in 2003, but it was a very, very blurry concept nonetheless. Now, we could define it, and we did define it. We knew it was going to involve using genomic information about an individual as part of their clinical care for prevention, diagnosis, and treatment. This morning we talked about precision medicine, which is a wider framing that includes as a subset of it genomic medicine, but precision medicine allows things other than genomic information to come in and be part of the same set of exercises to improve the practice of clinical care. But genomic medicine stays laser focused on genomic information. So there we were 23 years ago, completed the Human Genome Project, had an institute whose mission needed to change, and so it was going to change. It was going to change by making a major pivot. It had a new starting line, the Human Genome Project, and it had a new finish line, which we knew would take far more than 13 years and was going to be a little more vague, realizing genomic medicine. But like the Genome Project, it was going to be just another journey. It was going to be a journey that was going to involve many, many steps. Many of the steps we wouldn't know what they were until we got going. But once you get going, you take one step at a time, and every time you accomplish that step, you get to happily cross it off your list and move on to the next one, inching closer and closer and closer to your final destination. And just like the Human Genome Project, this was not going to be accomplished by one group, by one discipline, by one country. We knew it was going to not be a sprint, but much more analogous to a marathon. Multiple countries, multiple disciplines, many, many people running shoulder to shoulder for a very long time. But you just got to get going, and you got to just figure out what those steps are, and then start pursuing them one at a time. What NHGRI did, and you heard in the introduction from some comments Phil made, but it even predated my involvement in directly leading the strategic planning processes, was, but I was part of the leadership team along with Francis Collins, who was then the director, was to take basically the lesson learned from the Genome Project, which was that if you have a really big project, you use the community to help you formulate strategic visions on how to get the goals accomplished. Shown here are sort of the major publications that guided the Human Genome Project from beginning to end. And so the day the Human Genome Project ended, we had ready to go a publication in Nature describing the next phase of genomics research beyond the Human Genome Project. When you are successful in writing a good strategic vision and executing on it, they get outdated, so you have to renew them. And then by then, I was the director of the Institute by 2011. And then in 2020, in the middle of the pandemic, we had the latest version. And so these three strategic visions, I would argue, have been instrumental in helping not only NHGRI and NIH, but I would contend the world, sort of see a charting of the course that needed to be pursued in order to get us closer and closer to the realization of genomic medicine. Now, I've got 23 years of progress to report, and all of you appreciate it has been explosive in terms of the accomplishments that have followed. So I'm going to quickly go over some of the more proximal ones that are more on the basic to translational science so that I could feature a bit more what's relevant to this session, which is the genomic medicine implementation. So in witnessing what has happened over the last 22, probably it's now after the first year, probably could update this to 23, what have been the major steps and what did we do about them to sort of have them accomplished? Well, I would contend the first one is the most seismic. It's influenced absolutely everything. And it was, of course, the fact that that first human genome sequence produced by the Human Genome Project was incredibly valuable and incredibly expensive. It cost about $1 billion. We all knew $1 billion was too expensive for a clinical test, so we put in that 2003 strategic vision the idea of the $1,000 genome. That was the first time that was put into press, and guess what? We've now done it. In fact, we've surpassed it. We have reduced the cost of sequencing a human genome by more than 1 million-fold, reducing it below a $1,000 threshold, and we could cross it off our list. And the reason you have a meeting like this constantly talking about being overwhelmed with data is because of technology advances such as that seen with the $1,000 genome goals and what came out of them in terms of technology advances. That meant we had cheap sequencing. That meant we didn't have to sit and look at one reference genome sequence. We could go out and use cheap genome sequencing to sequence tens of thousands, then hundreds of thousands, and now millions of human genomes, catalog and make available to the world lots of information about the genomic variants of various flavors and forms, haplotype structures of various flavors and forms, and as best as possible, although still a lot of catch-up, getting as much diversity in those data sets as possible. But we have gone from having such cursory information about how genomic variation occurs across the human genome to now having incredible amounts of information about not only the points of our genome that are variable, but how they are varied and what populations we see them at what frequency. And so I think we can contend that this step has been not completed, but by certainly means we've made a tremendous amount of progress. But of course, just knowing about genomic variation is certainly not enough because you want to, of course, know how does that variation influence genome function, if at all. And while we knew this was going to be a seismic effort of interpreting those 3 billion letters, I would contend we've come a long way. We've profoundly advanced our understanding of how the human genome functions. But boy, we have a long way to go. Yes, we have a good inventory of human genes, but boy, we don't know what most of them do. And yes, we know that non-coding DNA is incredibly important, but boy, we have a lot to learn about the language of non-coding DNA. And as we heard from a talk this morning already, wow, there's this stuff called RNA, and it actually does more than just serve as mRNA, and in fact, it's medically relevant. Yes, there's a lot more to learn, but we've made quite a lot of progress. So it doesn't deserve a big check mark, but I'm going to give it a medium check mark, but it would encourage all the young people in the audience, keep on analyzing the human genome. We have a long way to go in interpreting the 3 billion letters, but we're making progress, and we need tools of both experimental nature and of computational nature to keep getting us a more layered and nuanced view of all the functional complexities of the human genome. And we've taken these tools and information about variation and growing information about function to significantly advance the unraveling of the genomic basis of human disease. Once again, I think we should say it's a medium check mark. Boy, we have a long way to go, but let's celebrate our triumphs. And what better place to start in celebrating our triumphs, which you've already heard about at this meeting, is in the arena of rare genetic diseases. Now, you younger folks may not appreciate this, but let me remind you that of the roughly estimated 10,000 rare genetic diseases that we think exist, when the Genome Project began, we didn't know the genomic basis of very many of them. You may wonder, how many did we know? We certainly knew sickle cell, and we knew PKU, and we knew CF. It got under the wire by just a few months. But in total, in 1990, there were only 61 rare diseases that we knew what the gene was that was mutated. But through all of the efforts that have been made, including efforts that NHGRI funded and other funders around the world, and you heard about the Gregor Consortium, and prior to that, the Centers for Mendelian Genomics, by now, 2025, it's over 6,000 rare genetic diseases. We now know what the mutation is. And yes, we have more to go, and yes, it gets more complicated, and yes, it keeps Ann up at night constantly thinking through the really hard cases that Gregor can't seem to solve, but we'll get there. And I just think we should acknowledge this is incredible progress. And of course, I fully acknowledge that the more complicated disorders that are multigenic, the common diseases that have a genetic complexity and a lot more influence in the environment. Yeah, GWAS has gotten us quite a lot of information. We can argue about how much more we can squeeze out of that and what the next steps are, but we've made quite a lot of insights, even through GWAS studies, and from that have come all sorts of opportunities, such as polygenic risk scores, which I'm going to say more about, and so forth. And yes, there have even been some triumphs. But ahead of us, we need to keep pushing on these rare diseases that we still need to figure out, and of course, lots of work on understanding the genomic basis of more common genetically complex disorders. But what about actually changing the practice of medicine? What about thinking about how genomics can be moved into the precision medicine armamentarium? And I would contend that we really are starting to see vivid examples at a time where, to be honest with you, some people didn't think it would happen this soon. Only gets a small check mark, only the tip of the iceberg, but we have gotten to the point where you can sequence a patient's genome, you can compare it to a reference genome sequence, you can make a list of those genomic variants, you can come up with a DNA report, and you can go off and implement genomic medicine. And I will admit, this is an oversimplification. I'll come back to this slide later, but fundamentally, this is a pathway that has yielded a number of areas where genomic medicine is now implemented. So I'm going to tell you what I think are the areas. I'm not going to go into any detail on any one of these. I'll just say a little bit more about certain ones. There are people in this audience who have far more expertise than I, but I regard this highlight reel of examples as being so gratifying, considering the fact that there were so many skeptics who even as recently as 10 and 15 years ago thought we were decades away from actually using genomics in medicine, and that has proven not to be the case. So the areas where I think genomic medicine is certainly here and now already, even though many more to come, there's some of the obvious ones that all of you appreciate. Cancer genomics, I think, will always lead the way. I always say that I think the greatest impact of genomics in clinical medicine is going to be in cancer. This is a sophisticated audience. You all appreciate the fact that genomics, or that cancer is fundamentally a genomics disorder. And there's so many things I could say about how it's changing diagnostics and therapeutic development and stratification, et cetera, and I'm not going to, because I think all of you are familiar with generally those things. I will just pick one example of a paper that came out, actually now it's a couple of years ago or a few years ago, celebrating the 15th anniversary where they had first sequenced an entire cancer genome. And they were just reminiscing about how much has changed from 2008 until 2023. And they just said that today, genomic sequencing as part of clinical care has transformed cancer diagnostics, clinical trials, and the use of new therapies to improve human outcomes for people with cancer. Our unprecedented view of the cancer genome empowers clinicians, computational biologists, and bench scientists alike to define biologically relevant groups of people with cancer, direct genomic inquiry, and ultimately identify new therapies and biomarkers. It is a field in and of itself with an incredible trajectory of accomplishment that has already been seen and will continue to be realized. The second area, which we've certainly heard quite a bit about even in this morning, in the arena of rare genetic disease diagnostics. Again, if you would have told me as a freshly graduated MD PhD student in 1987, training in laboratory medicine, that in my lifetime we would go from being able to have a patient in front of us with a developmental delay, a heart abnormality, and some suspicion of a genetic disorder, and actually sequence their genome and actually come up with an answer, I would have said never in my lifetime. And then if you would have said, oh no, it'll actually happen, while you're still around and you'll actually be involved, and I would have said, I can't even imagine this is even possible. But all this has happened, and it's happened so much faster than any of us ever anticipated. And this was actually the paper that was mentioned earlier in Ann's talk, where the diagnoses come up something like 30 to 50% of the time. The number is going to get better. We all can appreciate it. And with long reads and various other things. And the fact of the matter is, it's not even about just being like remarkable and anecdotal cases. I mean, there's even review articles that talk about the current state of implementation because this is getting implemented wide and far. Although, again, lots of things still need to be done to make this completely routine. But this is here and now. This has actually become mainstream, which is really pretty remarkable. Now, coming out of this have been all sorts of different areas where the rare disease diagnostics has become so important. I'll just highlight a couple that were pretty instrumental under my directorship of NHGRI. One was in the arena of undiagnosed diseases. Shown here is Bill Gall in the middle, who created the first undiagnosed diseases program within the intramural program of NIH, and he and I worked tirelessly to get this off the ground. When it was successful, it eventually then got to be nationalized under the umbrella of the Undiagnosed Diseases Network, and Anne even presented a case from that network earlier today. And now there's undiagnosed diseases programs all around the world, and Bill Gall travels all over the world, constantly helping other countries set these programs up. Another niche area of genome sequencing for diagnostics that I'm particularly proud of was the fact that early on as the NHGRI director, I hosted a workshop that asked the fundamental question, what might we be able to do with genome sequencing of newborns? Thinking mostly it was going to be about healthy newborns. But one individual, Stephen Kingsmore, came up with a novel application that we decided to fund, almost as a rogue application, where he proposed doing this to acutely ill individuals in the neonatal intensive care unit, but doing it rapidly. Well, the rest is history, because it turned out to be an incredibly successful endeavor. It has grown up considerably ever since then. Adoption has been growing ever since, to the point that by 2024, there's actually review articles that are written about doing rapid genome sequencing of acutely ill newborns, increasingly those in pediatric intensive care units. And so in this review article, they had 44 studies of children in ICUs with diseases of unknown etiology, 37% of which received a genetic diagnosis, 26% had subsequent changes in management, and net healthcare costs were reduced by about $14,000 per child. In 5 years, there's potential for infant and childhood mortality in the US and UK to have been reduced by several percent through the use of rapid genome sequencing as a first-tier standard of care test for children in ICUs with diseases of uncertain etiology. Again, I could have never predicted this was going to happen as fast and as widely as it already has. And it's, again, only going to get better. Some of you may have seen this breaking news from a couple of months ago, I guess it is at this point. We're at the Broad Institute at the Broad Clinical Lab. And you may have read about it in the New England Journal, where they really tried to move this faster, and they set a world record. Let me blow up that part where they talk about whole human genome sequencing analysis was completed in less than 4 hours, surpassing the previous benchmark, et cetera, et cetera. Pretty impressive that you were able to do that and actually get a diagnosis. And what I think is also interesting is, don't think this is just from the pediatric period. I don't know how many of you saw this paper that came out of the University of Pennsylvania, where they looked at a series of individuals in the adult intensive care unit. They excluded individuals who were there for trauma or for poisoning or for some known reason, and just took the people that, why are they there? They shouldn't be this unhealthy and be in an intensive care unit. Guess what? A quarter of them had a rare genetic disease. A quarter of them, and again, the number was probably higher once we get better at doing the diagnostics here. And so it really applies maybe to all patients in all intensive care units probably would benefit from having a rapid genome sequence. So I amplified this, but I go show this figure because rare disease diagnostics really is relevant across the lifespan. I told you about routine diagnostic circumstances, including for undiagnosed cases. I told you about rapid diagnostic genome sequencing. There's also lots of attention to reproductive genetic carrier screening for couples who want to or in circumstances where genomic information could be valuable. And of course, in a bit in my talk, I'm going to talk about healthy newborn sequencing, so hold that. But this is across the lifespan where rare genetic disease diagnostics is becoming increasingly relevant. And that's all after birth. What about before birth? Well, that leads to my third area of genomic medicine implementation, which turns out to be the #1 genomic test worldwide today, and that's non-invasive prenatal genomic testing. I bet most of you understand this, but let's just quickly, by way of review, for many, many years before genomics, we would look for aneuploidy through an invasive procedure like amniocentesis or chorionic villus sampling, looking for circumstances such as 3 copies of chromosome 21 or trisomy 21. That was then, but this is now. Now, we don't need to do that anymore because it turns out the placenta naturally sheds a small amount of DNA, which represents fetal DNA, into the maternal bloodstream. It's the minority of the cell-free DNA in the maternal bloodstream, but through a non-invasive, simple blood draw, and for a very reasonable cost that insurance companies are picking up and paying for, You could basically do the same kind of screening by simply counting reads, assigning them. And if you see a few too many chromosome 21 reads, you want to have a follow-up and you want to look more carefully to see whether that unborn child has a trisomy or some other chromosomal abnormality. The point about this is that the circumstance is such where the uptake has been so significant that it is now estimated that between 7 and 8 million pregnant individuals will get this non-invasive prenatal genetic test done this year, making it by far the #1 genomic test in use today. What about area number 4? Well, area number 4 is pharmacogenomics, and here I have to admit, there are some serious pharmacogenomic experts in this audience, including the person who introduced me. So I'm really going to give a very quick flyover, pointing out how everybody responds differently to all sorts of things, including roller coasters, but particularly to medications, where they all work, they just don't work for everybody. And for so long, we've known how imprecise we really are. This is old data, but these are 10 medications. Many of them are still in use. For every individual in blue where the medication works, or all the individuals in red where it didn't work as well as it should have, or really was problematic. But of course, the idea is to get the right drug for you, not by developing the drug, but by picking it from a list. Now appreciating the fact that embedded in our genome sequences are variants that are influencing drug metabolism, which in many ways influences whether you're going to be a good or a bad responder to a medication or need a different dose. And so the idea, which has had, again, the leadership in this room in terms of pharmacogenomics has been instrumental in making pharmacogenomics a very clear, important part of medicine going forward, is stratify, stratify, stratify. Take individuals of different genetic backgrounds and figure out which ones are going to have the bad responses, who are going to need a little bit different dosage, but find the ones that you could optimize relative to the medication. So one size does not fit all. You stratify, you stratify, you stratify, and then you just get the right drug to the right patient at the right dose. And needless to say, this isn't hypothetical, although the uptake has been slower than any of us had hoped for. You have companies, I'm not endorsing this company, that are offering all sorts, and many companies are offering this for many different genes, that influence many different drugs. Here are some representative examples, by no means a comprehensive list. But you could see this is relevant and it's real, and the uptake has been slower, and lots is being done to try to improve that, and we could talk about why it's been slower. But nonetheless, I think it's really important to appreciate this is still a growth area and one that we can expect lots more development in the coming decades. I also point out it's not just for adult conditions, and I just throw this article that's fairly recent with a nice infographic, and I think it's really important that the more we learn about pharmacogenomic options in the pediatric period, it really might drive this forward, especially thinking about newborn sequencing, which again I'm going to talk about in a couple minutes, but I just wanted to throw that out there. And last, these first 4 examples all relate to medicine, but what about prevention before we ever get there? And I do think genomics should be proud of some of the accomplishments that are coming out in terms of prevention. And in particular, I would point out that in the case of rare genetic diseases, it's well established that there are good prevention pathways, whether it's for this gentleman whose sister might have been diagnosed at a very young age with colon cancer, turns out to have Lynch syndrome. Now, through cascade screening, we can screen the whole family and try to identify through a preventive mode which individuals are likely to develop colon cancer and therefore should have higher surveillance than other members. Or this little girl might have been diagnosed at a newborn screening stage with something like PKU. We do a diet adjustment, and prevention therefore has a major impact on improving the quality of life of that little girl. What is exciting, and what has been discussed in multiple talks at both days of this meeting so far, I heard some of it in the workshops yesterday, of course, so for rare genetic diseases, it's here and now, but what all of you are looking, not all of you, many of you are looking at, of course, are polygenic risk scores. And here I think we are at sort of maybe a transition point. I was pretty firm in believing that it's under construction. It's not ready for clinical implementation. And I still believe that. We have to be really careful. And to further convince myself where we stood, over the holidays, I did some, which every once in a while I think is really worth doing, is just do some Google searches as to what's out there that people who are doing their own research on the internet are finding. And you are finding many, many direct-to-consumer companies offering polygenic risk score testing. Now, some of this stuff, maybe be fine, but again, because it's direct to consumer, you only have to go a couple clicks in to realize why I get so cautious about this. So this one looks innocent until you say, hey, I'm interested in this test, and then they start offering you other things about DNA testing for nutrients and personality and so on and so forth, and immediately you know you're getting into things that we really, as a scientific community, don't want to be endorsing. This company, once again, seemed innocent enough until you get a click in further where you find out DNA testing for weight management and mindfulness, whatever that means with respect to DNA testing. And the same with this one, where there's all sorts of things for slowing your aging and training smarter. And I can't even read all that, but your superpowers, that wasn't about it. Find your genetic superpowers. That scares the heck out of me. On the other hand, when there's a validated, reputable place, such as in breaking news from a few months ago, again, going back to the Broad clinical labs, it's much more measured. They talked about how they are starting to offer this as clinical testing for 8 conditions, very cautiously, and with a lot of good evidence behind it. And so I sort of feel like right now we can start tipping our toes in the water, just lightly, but again, I want to see it done at validated labs where clinicians are involved, as opposed to some of the direct-to-consumer. So we need to tread cautiously, but maybe we will see some progress in this arena in the coming few years. And so what all this I've quickly romped through over the last 15 minutes or so is this pivot where we went from the vaguest idea of what genomic medicine was going to look like, and now 23 years later, it's pretty good. It's clear, it's not done. In fact, we have far more to do than we've done so far, but it's brought some clarity. We have real examples, real challenges, and real clear path of what still needs to be done to make this broader. But that then brings the question that I'm frequently asked, and that question is, what is the next big thing in genomic medicine? It's a great question, and many of you might have different answers. I actually think that the answer, what I'm excited about in particular, it's just one person's opinion, is a seismic expansion of an established thing. What do I mean by that? Well, it really dates back now 60 years to newborn screening. And I think many of you are familiar with this, but let me just let this video run if you didn't know that in most countries, especially developed countries, within about a day or two, a child is subjected to this tortuous thing where they stick a little needle in their heel and get a little bit of blood out. That blood gets put on a little a piece of filter paper called a Guthrie card, and it goes off to the state lab. And what's happened in the 1960s and 70s, and then fast forwarded to 1990, 20, is that the state labs, public health labs, have gotten very organized at doing this, each one doing it a little bit differently, but screening for some number of genetic disorders. I actually looked it up this morning, since I'm here in Hawaii, 33 genetic disorders for the state of Hawaii, which is pretty average. Some are as high as 50, 60, but pretty much that's sort of in the bulk of where a lot of them are, around 33 or so. But of course, that's for 33 genetic disorders, and I just told you we now know the genomic basis of over 6,000. And so it really has always begged the question, and early on there was talk about this, even when the Genome Project launched. Would we one day, in a Gattaca-like way, which scared the hell out of people, was the idea, would we eventually sequence genomes of all healthy newborns when all the fruits of the Genome Project are realized? When the Genome Project ended, it started getting a little more attention. I even told about an early study we did shortly after I became the director. But we have all the infrastructure there. And the cost of sequencing has gotten lower and lower, starting to approach the cost of even doing the more traditional screens. And the idea of doing genome sequencing is once again, of all healthy newborns, is once again getting attention. Now, in the intervening time, others have pointed to this. I'd point out that Time magazine every once in a while likes to put out a futuristic essay or a futuristic issue. And they talked about now, I guess it's, now 12 years ago, they talked about by 2025, which was last year, everyone would get their DNA mapped. I might do a slight edit here. I think they meant sequenced at birth. Well, that was a little bit overly audacious, but here we are now in 2026. And what I will tell you is there is a lot of momentum growing. And there are major review articles and lots of discussions. And I will tell you that this is being really featured in many, many places around the world, and including here in the United States. I could tell you about study after study that's now being published, where this is really being looked at in various pilots in very serious ways. There's major review articles being written about this. This one was in Clinical Chemistry by Wendy Chung last year, and I just got an alert this morning that on the very first issue of the journal Clinical Chemistry that just came out on page 123 is another review article talking about genome sequencing of newborns. Had a very similar title to that, but now it's 1 year later. And the reason there's all this attention and these reviews and these articles is that all around the world, pilot studies of sequencing the genomes of healthy newborns are popping up. Every one of these pins in this world map represent one of these major studies. All this has now been organized under a consortium called the International Consortium of Newborn Sequencing, or ICONS, which is attempting to sort of bring these groups together to have meetings and strategize and working groups and so on and so forth. Among those various pins on that world map include a major UK study called Generation, a major US study under Wendy Chung's leadership called Guardian. And then the UK study has gotten so much enthusiasm in the UK that last year they just declared that in the next 10 years they expect every newborn in the UK to have its genome sequenced at birth. And here are various articles talking about that. So the UK has really put down the marker and said this is where we are heading, and they've even given a timeline for it. So it's not surprising there's yet more breaking news with new studies, whether it's Thailand, new studies, whether it's Denmark, or new studies that just got launched, whether it's the United States. And here, in a new program that I actually had a little influence on before I departed NIH, the Beacon study, which is now looking at partnering with the state public health labs and running with it to try to generate, as a pilot, what this might look like to do genome sequencing as a screening modality in the same spirit of the newborn screening methodology or approaches that have been used now for decades. And so this is really getting exciting and a lot of attention. I want to just point you towards a TED Talk. I'm not going to, if you have 10 minutes, Robert Greene, we share the same last name, but we are not related, as far as I know. But he did give a very good TED Talk, and I just want to play 1 minute clip because it actually speaks to some of the things that many of you were thinking about, and Robert's very articulate, so I find this to be a very entertaining clip, and maybe you'll want to go watch the whole TED Talk when you have 10 minutes.
But if we really want to invent the future, we've got to do something different. If we really want to invent the future, we've got to realize that a child's DNA doesn't change over time, but the science is changing all the time. And so what that means is we should sequence your child's DNA, and we should revisit and reanalyze that DNA over and over again to truly create the dream of genome-informed medicine. Because each and every year there will be new insights and new treatments available. Well, this isn't offered anywhere in the world, but I'm happy to tell you that we are trying to build this. We are building an AI-enhanced digital health platform so that you, your grandchildren, your children, your pediatricians, your healthcare centers, your employers, your nations can do this at scale. It's going to take a certain amount of courage to change the way we think about disease, to embrace the knowledge of risk in order to preserve our health rather than waiting for us and our children to get sick and treating them there. But if we can do this, if we can embrace this, we can save millions of lives and usher in an entirely new era of genome-inspired medicine. Thank you.
And, you know, some people have said, have I come out on apology or my views of this? In 2021, my colleagues, a few colleagues of mine at NHGRI, we actually basically came out and talked about this. Our theme at that time was it's not a matter of if, it's a matter of when. I think if we wrote about it now, we would have given a more aggressive timetable than we gave back then. We hedged a little, but a lot has happened now in 4 and 5 years. And I think what we really need to be doing, and I'm spending a lot of my time now trying to think about this and trying to help the systems do it, is to prepare for a future like this, where children are going to come in with genome sequences available. However, we've got to be really careful. And we've got to stage this just right. We need to learn from these pilots, because as happy as this family looks, if we overwhelm a family with a new baby with a lot of genomic sequence information, we could really have a situation that looks like this. And so this is the challenge I think we all need to face. And there's a lot of bioethics that has been talked about for a long time, such as that Time magazine cover from 2012, but more recently looking at New York Times and various things that our ethical, legal, and social implications research program at NHGRI have been looking at and will continue to look at. We need to be thinking about the bioethics very carefully along the way. So, genomic medicine has arrived, and we should celebrate, and it's wonderful. But let me just end with a few more slides that really point out, I don't want any of you to come away from this talk thinking this was simple, this was linear, or that it didn't have a lot of twists and turns and at times moments of outright exacerbation of concerns about where we're going to be successful. Because research, it really is such a journey of twists and turns and constantly facing challenges. And we always need to be thinking about those challenges as we go forward. So I could give a whole nother hour talk, which I won't, about the challenges, but let me just touch on two, just to show you that I'm being very realistic and balancing that while we've accomplished a lot, we still have major things to deal with. Long list of challenges, I'll just highlight two. One, which was very nicely illustrated in today's session, is that clinically understanding the patient's genome sequence is not simple. Everybody, many of you talk about the highlights, and they are incredible triumphs. But the fact of the matter is this slide was a gross and profound oversimplification. Chances are, in many cases, when you genome sequence a patient and round on that patient, most of those variants, you don't know what they mean. And what we are doing now is basically cream skimming to the best of our ability, but we must acknowledge we need to get better. And many of you are involved in fixing this grand challenge that will continue to be very, very pivotal for the use of genomics and diagnostics. But we also need to be thinking about the fact that we now are part of healthcare in the genomics world and the precision medicine world. And that comes with some pretty big responsibilities. So if I go back to when I got involved in genomics, it was fun, it was nerdy, and we didn't have to worry about a lot of things. It was sort of, since we're coming off of the holidays, it was sort of like being at the kid table during the holidays. We would nerd out, we would geek out, we talk about mapping and sequencing, and we didn't have a care in the world. But you know what's happened? Now that we're touching healthcare, it's no, Eric, you can't sit at the kids' table. I don't care that they're talking about iPhones, Instagram, Taylor Swift. You have to stay here and talk about mortgages, life insurance, taxes, politics, and genomic medicine and healthcare. That's what it feels like, because the good news is we're touching healthcare. The bad news is that healthcare is fraught with adult problems, forcing us to the adult table, where it's not much fun, but it's the mature, important thing to do is to think about our place in society, and nothing is more complicated than healthcare in society. And so we are dealing with challenges of how does this get delivered, and how does it get paid for? How do we regulate these new genomic opportunities and tests? Obviously, many issues about equity that sometimes hit major political headwinds that we then have to deal with, such as what's going on now in the United States, genetic privacy issues, and obviously genomic literacy for our patients, for our healthcare professionals, not just the physicians, all healthcare professionals. All of these we need to be thinking about, and we certainly are, and will continue to be really tackling them head on. And of course, it's also an issue of many other challenges, which I'm not going to touch on, but many of you are fully aware of. So I hope what I have done is to romp you through these years of progress, where we went from a double helical structure, used the Human Genome Project to put a magnifying glass and be able to read out the letters of the human genome, and then ever since then used the threads of genomics to weave into this growing tapestry of genomic medicine, precision medicine, human genetics, so many areas of data science and AI, and including continued weaving of this into the basic fabric of healthcare. And this has come along with this incredible growth of a community that you are all now a part of. And again, it started with just a very small group of pioneers working on the Genome Project. And just think about 35 years later, how big our community is and how interwoven they are and standing shoulder on each other's shoulders to sort of realize things that we never thought was possible in our lifetime. And I'll just end by pointing out, I have nothing to do with anything beyond diagnostics, and NHGRI, when I was there, and even still now, really not into therapeutics. But holy cow, are there some spectacular new treatments that are coming out because of these advances that we've put forward in terms of gene therapy for disorders, in terms of gene editing, in terms of RNA therapies, in terms of enzyme therapies, in terms of gene and cell engineering therapies. And needless to say, I think it's incredible that we're able to keep this progression going even beyond genomic diagnostics. So I am standing between you and lunch, but I hope you'd enjoy this story. Oh, and I missed small molecules because it was the bottom. So I will stop there. I'm happy to take questions, but I also appreciate that it's lunchtime, and I'll be around if people want to just stop me anytime.